Hello, everyone. Welcome to today's live broadcast, Single Particle ICP-MS and its advantages in analyzing nanoparticles in environmental matrices. I'm Laura Bush, the Editorial Director of Spectroscopy, and I'll be your moderator for today's event. We are delighted to bring you this web seminar presented by Spectroscopy and sponsored by PerkinElmer. PerkinElmer is a global leader focused on improving the health and safety of people and the environment through the development and delivery of technologies, services, and solutions that are used in critical applications. From environmental monitoring, water analysis, and food safety to clinical diagnostics, drug discovery, and biomedical research, PerkinElmer is committed to advancing science, collaborating with their customers, and putting innovation into action for a healthier today and an even better tomorrow. We have a few important announcements before we get started. The webcast is designed to be interactive, so we encourage you to ask questions. You can submit your questions by typing them in the Q&A box, and you can find that by clicking on the red Q&A widget at the bottom of your window. You can enlarge that slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically. And if you have any technical problems viewing or hearing the presentation, please click on the question mark Help widget in the dock at the bottom of your window. Before we get started today, we have two brief polling questions. To answer, please click directly on your screen. Here is our first question. What market segment are you a part of? Academia, government or regulatory, industry, or other? I'm sorry, the last option should say D, other. So again, what market segment are you a part of? Academia, government or regulatory, industry, or other? I'll give you just a quick second. Click right on your screen, and then we'll move on to our next polling question. Okay, and here's our second question. Again, click right on the screen to answer. What is your primary application of interest for nanomaterials? Environmental, such as studying the fate, bioaccumulation, bioavailability, or toxicity. Biomedical, for nanomedicine or nanobiotechnology. Industrial, for surface coatings, textiles, nanocomposites, cosmetics, etc. Or for energy, such as fuel cells and batteries. So again, what is your primary application of interest for nanomaterials? Environmental, biomedical, industrial, or energy? And we'll give you just another second there, and then we'll move forward. Okay. I would now like to introduce today's speakers, Chadi Stephan, Kevin Wilkinson, and James Ranville. Chadi Stephan is the Manager for Global Applications of Nanotechnology at PerkinElmer. He received his PhD in Analytical Chemistry from the University of Montreal in 2008. Dr. Stephan has worked as a project manager for QSAR risk assessment services before he joined PerkinElmer as an inorganic product specialist, supporting the inorganic business. Over the past few years, his main research activities at PerkinElmer have been in developing single particle ICP-MS. Kevin Wilkinson is currently a professor in environmental chemistry at the University of Montreal, having started his career at the University of Geneva. His research is aimed at gaining a molecular-level understanding of contaminant bioavailability and mobility. He is currently participating in numerous team grants on the fate and bioavailability of engineered nanoparticles in the environment. Dr. Wilkinson has more than 130 publications and over 5,200 citations to his work with an H-index of 42. He has recently established a world-class analytical laboratory specializing in the characterization of nanomaterials, called the Center for the Analysis and Characterization of Engineered Nanomaterials. Jim Ranville is a professor in the Department of Chemistry and Geochemistry at the Colorado School of Mines. He received his PhD from the Colorado School of Mines in 1992. His research has focused on the environmental fate and aquatic bioavailability of trace metals, including metal-containing nanoparticles. Dr. Ranville has been very actively involved in the development of single-particle ICP-MS for environmental applications. Thank you all for joining us today. Chadi, please go ahead and get us started.
Thank you, Laura. We're going to be starting over this webcast entitled Single Particle ICP-MS and its Advantages in Analyzing Nanoparticle Environmental Matrices. On the agenda for today's webcast, I'm going to start introducing what Single Particle ICP-MS is. I'm going to go over how it does work and what are the measurements that this technique will allow us to be able to measure. I'm going to shortly glance over how we acquire the data in an automated way using the Synergistic Nano Application Module. After that, I'm going to turn it over to Kevin. He's going to be talking about the fate of nanoparticle in environmental matrices, talk about the different available analytical techniques that allows us to investigate or track what happened to nanoparticle in environmental matrices. And of course, he's going to be talking a little bit on the advantages of single particle ICP-MS when it comes to measure few nanoparticle in an environmental matrices. After that, we have Jim. Jim is going to be talking about tracking the transformation of nanoparticle using single particle ICP-MS. He's going to be talking about the microseconds dwell time and how it is advantage in complex matrices. And after that, he's going to be touching base on the ability of using particle by particle at a multi-element level using single particle ICP-MS. All of that is going to be concluding by tracking the bioaccumulation of nanoparticles in different bioorganisms using single particle ICP-MS. To get you started, I would like to take a minute and discuss what is nanomaterials or smart materials or nanocomposite. And as you see, this is one of my favorite slides because it summarizes and it puts everything in a visual aspect. Nanomaterials or nanocomposite are nothing than ongoing current materials that we currently use on a day-to-day basis, from cosmetics to food containers to high capacity processors, high efficient batteries, drug delivery systems, and cancer treatment. They're already products that we are currently using. And this is really due to the engineering at the nanoscale. Now, why it's important and why it's important to understand nanomaterials. The nanomaterials, because have a very high surface reactivity. They do behave differently. Thus, we have to advance our research to be able to follow them, track them, understand how do they react with our environment. Or even when we are looking at a production level, we want to be able to improve the quality control of our production. We would require analytical tools. For today, we're going to be looking a little bit about the environmental release. If we follow the graph from manufacturing, starting with the raw materials to the manufacturing, to the usage, to the end of life, eventually, these materials, engineered nanomaterials, are going to be released into the environment. Thus, we need technologies, we need technique that allows us to track, to better understand what happened. As Laura mentioned, Kevin is investigating it at the molecular level. Jim is looking more at what happened. How can we differentiate anthropogenic release from natural release of nanoparticles? And this is what we're going to be discussing during today's webcast. To achieve that, this webcast is part of a series of 3 webcasts. The first technique we're going to be discussing today is the single particle ICP-MS, and it's going to allow us to look at metallic nanoparticles. It's going to give us the composition, the particle counting, and we're going to be able to track the dissolution and the aggregation of nanoparticle using this technology. However, when we are looking at characterizing nanomaterials and nanoparticles, we want to be able to understand lots of information that evolve around these nanomaterials, from what is sitting on the surface, what's the surface reactivity, do we have organic molecules on the surface? And this is where we're going to be touching base on that technology using our second webcast, where we're going to be talking about the DSA-12 and its ability to look about the organic layer sitting on the surface of nanomaterials. As for the third webcast, we want to be able to look at the production, the quality control of nanoparticles, and how can we use a UV-Vis near-IR technology with a different integrated module in it that allows us to expand the field of better understanding the production and the manufacturing of nanoparticles or nanomaterials. For today, I'm going to be concentrating on single particle ICP-MS. And what is single particle ICP-MS? If you ask me this question, I will tell you that it is a new advancement in ICP-MS that allows us to look at individual inorganic nanoparticles. It is one of the techniques, the only techniques that is available that allows us to differentiate between an ionic signal and particulate signal in the same sample without any prior separation or any prior sample preparation for that sample. It is an element specific, provide us the composition of the nanoparticles. It allows us to look at their sizes and the size distribution. It is a very sensitive technique, and when I say very sensitive technique, it allows us to go to the sub-ppb level when it looks at an element composition. But when it comes to nanoparticles, it allows us to look at few particles in the solution. And this is one of the key advantage when we are using single particle ICP-MS to look at nanoparticle release or natural release in environmental matrices, because we're not going to encounter a large number of particles over there. So we wanted a technology that allows us to go and look for those few particles in that matrix. It is a technology that's going to be measuring the core size of the nanoparticles. So it's a mass-based technology. It does measure the core size. It does not measure the hydrodynamic diameter. So it does not measure the overall size. It's really concentrated on the core size. And it is a technology that allows us to track the nanoparticle dissolution, because it has the ability to look at both fraction, ionic and particulate. And we'll be able to track agglomeration as well. So in the cartoon aspect, this is how the single particle ICP-MS works. So we're going to be aspirating a solution of nanoparticles. As the particles go through the plasma, it's going to create an ion plume or a pulse. Mainly it's a cloud of ions all condensating going together. And when this cloud of ions get to the detector, we're going to be getting a spike. And this spike is really the science behind the quantification of the size of the nanoparticles. So, as I mentioned a slide ago, the single particle ICP-MS is an extra dimension to the ICP-MS itself. It's because we added time at an extra resolution first. So let me guide you through what we can do on single particle ICP-MS. We will be acquiring data consecutively, so it's continuous data acquisition. We can do go down as low as 10 microseconds. But from a scientific perspective is that we're constantly looking, so every particle that goes through the plasma is actually seen and counted. So the number of pulses is related to the particle concentration. And the intensity of the pulse, because now we are acquiring all the ions in that pulse, that amount of ions is related to the particle mass. And of course, the background, which is the straight line on which we had the pulse that comes on top of it, is really the baseline. So it is equal to the ionic fraction or the dissolved fraction that is in that sample itself. Now, from a mass perspective, when you are using single particle ICP-MS, you do not need particle standards for the particle that you are measuring. All you have to do is basically use the same ionic standards that you use on a day-to-day usage in your ICP-MS. You're going to be calibrating the instrument using those solutions, and you're not going to calculate something called the mass flux. We're actually calculating the response of an instrument based on time for the amount of ion that she's going to receive. And this is what then allows us to convert a particle signal acquired over a certain amount of time, quantitate the ions in it, and correlate that to an amount of mass. And the formula to use for such is the following. You're going to have the mass flux, which is equal to the transport efficiency of the system or your instrument that you're using, the sample flow rate, how much we're introducing liquid in it, and the concentration. And that's going to give us the amount of mass by time. And we're going to use that to convert the intensity of the nanoparticles acquired over a short time, which is usually 300 to 500 microseconds. And we're going to be able to get the amount of mass generated from that nanoparticles. As we all know probably on the call that ICP-MS is a mass-based technology. Now we are getting the mass for our nanoparticle. If they are spherical, you're going to use the spherical conversion from a density equal mass over volume, and we're going to get the size of my nanoparticles. There's nothing to stop us of using the same technology to calculate the size of non-spherical nanoparticles. The most important thing is you have to know the size of the particles or the materials that you are analyzing using single particle ICP-MS. In this small movie, really I would like to show you what we can do from a perspective of real-time monitoring of nanoparticles. If you look at the graph on the left-hand side on the bottom, you will see spikes. These spikes are really the nanoparticles entering the plasma. So every time a particle goes into the plasma, it's going to generate a pulse. That pulse is going to be quantified based on the surface area, because we are able now to acquire multiple points in every individual pulse. We are using the surface area under the peak to quantify the size of the nanoparticles. If we look on the right bottom graph, it's a graph that has the frequency versus the peak area in counts. So the frequency allows us to calculate or to count the nanoparticles by amount of time. So we would know how many particles has entered my system. And using the peak area, we'll be able to quantify the size of that nanoparticles. So once an analysis is done, we have something that allows us, because now the ICP-MS is not just looking at ions and concentration. No, we are looking at a more complex system. We are using a technology that allows us to look at the ions and also at the particles. And in the majority of the system or samples, we're not going to have just one size distribution. For example, on this screenshot, we have two sizes. So we created something called the integrating window, and that allows us to zoom on the population of particles that we are interested in. And based on that zooming, we will be able to get all the good information related to that size distribution that we are interested in. So we'll be able to get the most frequent size, we'll be able to get the mean particle size, and as well we're going to be able to get the counting, and we're also going to be getting the intensity of the dissolved fraction. All those information could be exported to an Excel. Either you can export your whole design, or you can export individual sample by itself. With this, I conclude my section about introducing what single particle ICP-MS. I will be turning it over to Dr. Wilkinson. He's going to be talking about the fate of nanoparticle in environmental matrices. Kevin?
Great, thanks, Chadi. So as Chadi said, one of the things that's really interesting our group is the fate of nanoparticles once they're in environmental or biological systems. So you see in this figure, as if you throw a nanosilver in natural water, some of the nanosilver will dissolve into ions. Those ions can precipitate or be complexed by ligands in the water to form either stable complexes or precipitate. They can be absorbed onto other colloids. The nanoparticles themselves can be absorbed onto colloids or agglomerate. And so all these questions are really important to understand the system if we want to really understand the bioavailability and the mobility of these materials in the natural environment. Now, in our lab, we're using a number of different techniques to get at this information, get at these different sizes. And the point I wanted to make here was that all of these techniques are really vastly different in the principles, and then, of course, in the disadvantages and advantages. So you're really not measuring the same thing. For example, with microscopy, you're measuring the width or the height of the samples. As Shadi said, this ICP-MS is a mass-based technique. DLS and nanoparticle tracking analysis, you're looking at hydrodynamic diameter, Q-nano, volume-based, etc. So all these different techniques are giving us a fundamentally different signal. Now, when you're testing out these techniques before purchasing them, you always get this same response if you're working with a single monodispersed particle in solution, for example, a polystyrene. And the techniques at very high concentrations and with these monodispersed standards will generally all give you the same value. But if you look on the right-hand side here, in natural systems, we're usually in the presence of mixtures of dissolved species, nanomaterials, agglomerates. And so you get diameters, number-based, weight-based, and Z-based diameters that are all fundamentally different to start with. In addition, we're in these really complex matrices, and really a key point here is that we're expecting very, very low concentrations of nanomaterials. So I'm sorry, I went backwards there. So I wanted to show you this here in this first figure. This is really a simple figure. What we've got is a serial dilution of nanomaterials, the nano silver, and so what you see is you essentially scale the concentration of the nano silver, but the dissolved silver doesn't scale precisely with the concentration. So on the right-hand side, what you see the implications of this. The ratio of ions, or the silver ions to silver particles, will change as you decrease the concentration, or as you increase the concentration. So what this implies is that we can't just measure our stock solutions and then suppose or assume that we're going to have the same answer at any dilution or any concentration. That in this case, and this was done with 3 different analytical techniques, several different coatings, several different nanoparticles, we get an increase of dissolution or oxidation in the case of the nanosilver as we're going to lower and lower concentrations. So it's vitally important that we're measuring the nanoparticles or characterizing the nanoparticles under the exact precise conditions where we're interested, where we want to be doing the measurements. Here you see with a single particle another example of that. If we just spike a millicube water with 20 nanometer nanosilver, what you see here is with time, as you're going down the figures, you get a displacement of the peak to smaller and smaller particle sizes, and actually the counts go down very substantially as well. So we're interpreting this as being mainly due to dissolution of the nanosilver. If we look now in a natural water with this more complex matrix, well, what you're seeing is actually going in the other direction, or slightly going in the other direction, seems to be getting more polydispersed samples, probably related to agglomeration of the sample. We are seeing a decrease in the particle concentration, but you're getting a shift more to the right. So again, part of this problem is related to the matrix, and if you're interested in seeing a little bit more on the transformations of the silver nanoparticles, you can go into this application note that we produced a little while ago. But what I'd like to do in the rest of my time is to show you what we get when some of our newer results, some of the new work we're doing to try and get rid of some of these matrix effects. If you couple your ICP-MS now, or your single particle ICP-MS, with different kinds of columns. In our case, we're using hydrodynamic chromatography. This is really similar to FFF, where the retention is a function of particle size. Most groups are using FFF now, but it's really the same principle. You get retention times the particles coming out at different times based on their radius. The other thing that we're doing is coupling the ICP-MS to ion exchange resins. So, for example, to Helix resins, where you're removing the cations, to XAD resins, where you can remove a lot of the organic matter. You see here the resin in the figure on the right. So what that does, as Shadi explained to you earlier, we have a real problem, or one of the problems associated with single particle ICP-MS is really setting the threshold level, distinguishing between what's dissolved and what's particulate. In this case, on the left-hand side, it's the dashed. We've set the dashed yellow line to distinguish between the ions and the particles, and that dashed line is seen by the dashed vertical lines in the right-hand side. Now, based on where you put that line, you're going to get more or less particles in your quantification. So, what we've looked at is varying the dissolved concentration, which is essentially giving us a difference in the signal to noise, or the particles over the dissolved. And you see that here for a given intensity ratio, given signal to noise ratio. The more dissolved metal that there is in the solution, the worse off you are. The harder it's going to be to detect those really low concentration or really low sizes of particles. And so let me show you what happens when we do that. Well, we're trying this in single particle ICP-MS mode, and you see for a 20 nanometer silver nanoparticle, we can get a peak, but we're missing some of the information at the lower particle sizes. If we run this through a resin now, the ion exchange resin, we essentially remove that continuous noise or the background signal, and we can get a much nicer signal, clearer signal for the nanoparticle. This one's even a little nicer. We've got a mixture of 5 different nanoparticles, and you see by single particle ICP-MS, we can pull out 2 peaks from that when we're in the presence of all these other dissolved ions. But when you run this first through an ion exchange resin, there we again remove the dissolved, and you can really pull out all 5 of your nanomaterials. So we're really getting some additional information by coupling with these different columns. And a last example for that, this is a nanoparticle that we're generally not able to see by single particle ICP-MS. And again, the problem being that the dissolved metal is just too high. So our noise is very high compared to our signal. And you see the result of that. This is a 50 nanometer nano zinc oxide, and you see that you've got a few signals, but it really just for probably the large agglomerates or large particles in that distribution. When you run that through the IEC column, the ion exchange column, you can see that we are able to distinguish then our 50 nanometer. Now I've cheated a little bit here because we're also using these very low dwell times or short dwell times that Shadi was talking about. So that's doubly helped us. That's been able to really give us an additional benefit in getting to these really highly dissolving particles and probing some of the information. Now that was done in water, in millicube water. If we do this in natural waters, you'll see it's not quite so easy. In this case, on the left-hand side, we've run through a resin. The right-hand side, run through a Culex resin, and you see because there's so little free silver here, the Culex resin doesn't really help us a lot. Most of the dissolved silver, in fact, it can be seen in the next one, was bound likely to organic complexes. And that then by changing the resin, swapping out the Culex for an XAD resin, we can remove that dissolved, get a much better particle distribution, and also at the same time get some information on the nature of the chemical species in the solution. So finally, I just want to show you a couple applications with the hydrodynamic chromatography. Again, I told you or showed you that the retention times are really as a function of our hydrodynamic radii. You can just pump that directly into an ICP-MS, and the ICP-MS with its very low detection limits will give us really nice information on the particle concentrations, and by knowing the retention times, we can get the particle sizes. Now, in this case, the red, black, and blue curves were done with 3 different matrices, and so you see that, but with exactly the same peak. So you see that the retention times are not only affected by the nanoparticle themselves, but also by the matrix. So we have an additional advantage by switching into single particle ICP-MS mode. Not only will we get information on the concentrations, we get information on the particle numbers on the lower left, and some independent information on the particle diameters. So information on the particle diameters from the retention times, but also information from the single particle ICP-MS, albeit with slightly higher detection limits. So that's what I've got to say for now. I'd like to especially thank Majid Hadwi, Kim Prue, and Vladimir Medzen, who provided the results, and the funding agencies that are there, and pass it over to Jim. He's got some really interesting applications to show you from there. Thanks, everyone.
All right, thank you very much, Kevin. I'm going to continue the discussion on how single particle ICP-MS can be used to study environmental conditions under which nanoparticles are transformed. And I'm going to highlight primarily the advantages of using microsecond dwell times, reducing the dwell times down the order of 1/10 of a millisecond, and what that helps us do. The next slide is a bit busy, but it summarizes a lot of the processes which we hope our technology for measuring nanomaterials can capture. On the left, we point out how even during the product manufacture, pristine nanoparticles, those things that we've tended to work with in the past, are altered by incorporation into products and then weathering those products and then eventually environmental transformation. And some of those environmental transformation processes are shown on the right. You have physical mechanisms such as agglomeration, chemical degradation such as oxidation or other processes, surface modification where the surfaces are changed. For today's discussion, I'm going to focus almost exclusively on dissolution. How do we track these changes to the particles using single particle ICP-MS? Kevin already showed some slides that clearly show how dissolution can be an important process, changing the conditions or the characteristics of nanoparticles. In most cases, if we want to study the dissolution of a nanoparticle, say for example silver, one might focus on how do environmental factors such as DOC and ionic composition affect this dissolution by trying to measure silver ion release. Well, in many cases, as Kevin pointed out, you don't see much silver ion. It's transformed into dissolved organic matter complexes or other things and can be lost. What we propose is to follow the changes in the nanoparticle, essentially the dissolution, the reduction in size, and from that we can mathematically calculate dissolution rates, again, by tracking the particle and not trying to track the ion. We put together an application note a while ago, and from that application note we have the figures on the right, which simply show that in different media, DI water, tap water, local stream water, the rates of dissolution are varied. What's shown in the upper right is a series of distributions. These are still presented as particle size, where you can see our 90 nanometer particle at time 0 is shrinking in size. What's not really shown very well because we removed it is, as Kevin pointed out, the dissolved ion background is increasing. Kevin was quite successful in his ion exchange approach to removing that background, but another thing we can note is that as we reduce our dwell times, the contribution of the background relative to the signal can be reduced. So in this particular slide, we show a 10 millisecond dwell time, something we kind of started out working with when we first started with single particle. We've now reduced it to 0.1 millisecond dwell time. Notice that background peak on the sort of the left graph. It's centered somewhere around 100 counts or so. Of course, if I drop the dwell time by a factor of 100, I drop my background down by a factor of 100. So now my background's on the order of 1 or 2 counts. As you can see, as you go to the right, if we take the same amount of nanoparticle silver and increase dissolved silver, at 10 milliseconds, we quite quickly lose that nanoparticle signal in this large background. But with the microsecond, the 100 microsecond data collection, we still are able to resolve particles on these higher backgrounds. And as Kevin mentioned, you combine the two techniques, the ion exchange and the short dwell times, and you may improve this even more. Now, if we think about how we might tease a little more information out of this approach of single particle, let's think a little bit about the composition, or I should say the structure of the particles. If we combine some of these dissolution experiments with single particle ICP-MS, how might we learn more about how elements are distributed in nanoparticles? We're studying this by starting out with a fairly simple, I guess it's a complex material, but we're using a core shell particle. It has a gold core with a silver shell and a PVP stabilizer coating. Of course, ICP-MS doesn't show us anything about the PVP, but it does give us the equivalent mass of gold and silver in a particle. This corresponds well to what also the TEM sees. The TEM also doesn't really see the coating. But again, as Kevin mentioned, if you use FFF, you can get the hydrodynamic diameter, which is shown in the lower right. And that's a fractogram showing that, again, indeed, the gold signal, which is red, and the silver signal, which is black, coalesce in the FFF together. So this is not a mixture of a gold particle and a silver particle, but truly a core shell material. And of course, I know what this particle looks like, and so I can study the dissolution of this and look at how I might be able to differentiate shell versus core. But if I didn't know what this particle was made of, then maybe this approach would also give us more information about structure. So the approach we've taken as a sort of a proof of concept is to suspend these particles in millicube water and 1% aqua regia, collect the data with the Syngistic Nano application module, and track the changes in these particles to, again, get at structure. If we go to the next slide, what I've got is a series of slides that show what the particle size distributions, or at least the intensity distributions of these particles look like with time. So when we first prepare the particle, this is our silver distribution centered around 200 counts or so. As I step through these 30-minute time increments, you can see, as we expected, because the silver is exposed to aqua regia, it is dissolving and the peak is shifting. As we start getting very small, we start to see that the particle signal is actually being lost in the background, which actually has been subtracted. So you don't see the background. But by 4 hours, there's very few particles left. If you look at this plot comparing what happened to the silver in millicube water over 4 hours, there is essentially no change in diameter. But as you can see, the silver particle is shrinking with time. By the time you get to about 2.5 to 3 hours, you run into that little bit of issue of the background now dominates the signal, and you have to be very careful. This increase in size is not really a real phenomena, but we have so few counts, we're only detecting perhaps a few of the very tail end of the distributions. So let's compare that to the gold core. Now we've got a distribution centered around 40 counts or so, and as we step through our time points, what you notice is it's not shifting in size. The gold core is protected from the aqua regia, but we are seeing substantial decreases in the particle number. So this is really highlighting what we need to know if we're going to also look at single particle, not as just a sizing technique, but a particle counting technique. Again, because the gold is protected in both aqua regia and millicube water, the core doesn't shrink in size. However, I went too far. Go back. We can see that the gold core, the gold particles, because they're present with the silver, they both have to track each other. So in millicube water, both gold and silver decrease in particle number. In aqua regia, the gold decreases the same rate, but for an aqua regia, the silver particle has this more dramatic loss. So it's really highlighting there's two processes going on. Some type of loss of particles to the containers, our hypothesis, on top of the dissolution of the silver, which basically takes our particles into the background, so we essentially lose them. So I think this highlights two things. One is that using these types of more complex materials might help you in order to track what's really going on in your experiment. In this case, we might have thought we were just seeing silver dissolution, but in fact, we're seeing two processes. And also just the care that needs to be taken in, again, making this a particle counting technique. Now, the other issue I wanted to address, as Kevin mentioned, in complex matrices, not only are we going to possibly have our engineered nanoparticles kind of shown in the center and right of this image, but also we have at much higher levels natural particles that are going to form a background. Our technique has to be able to distinguish between these, and this is where the elemental composition can be useful in this approach, and that we get this from single particle, which we would not get from other techniques like DLS or nanotracking. So we're going to try to look at elemental composition and how that might help us differentiate this engineered nanoparticle from the background. So one thing we can focus on, and just take this as an example, is looking at cerium and lanthanum in the environment. I have a cartoon on the right that shows that if I have a natural particle, we might have both cerium and lanthanum ions in the particle. If we introduce that into the single particle and we could actually track both elements, we might see a signal, that pulse, that show both elements. In contrast, an engineered cerium oxide might be isotopically pure, and therefore we would not see any lanthanum, we would just see cerium. Now, if you look at the left, that's a data set from the European database that shows on the order of about 800 samples of stream waters that show there's a fairly tight distribution between cerium and lanthanum in nature. So again, we would expect that if we could look at this, the composition of the particle on a particle-by-particle basis, we might be able to start to tease out, to some degree, the presence of these two types of populations of particles. So the way we might be able to do this, and I'm going to advance one more slide, is think about now by reducing our dwell time to on the order of 0.1 millisecond, we might be able to detect both cerium and lanthanum particles as they enter the plasma. Of course, we have some missing data. We have the settling time, which hopefully we can keep quite short, that we have between our first ion and our second. So there's a bit of data loss, and we have to deal with that. But what we found is if we integrate over enough particles, a few thousand, we start to integrate out some of this problem of lost data. So for as an example, if you look at a local stream water sample, the raw data is on the left, and you also introduce cerium oxide into that sample. On the upper right, you can see for the most part, and this is a work in progress, but for the most part, we see cerium and lanthanum occurring together. And as we introduce cerium oxide into the system, we start to see more and more cerium-only pulses. Again, this is a work in progress, and further work needs to be done to look for what levels of cerium are really required for us to statistically be able to count cerium-only particles from those containing both elements. You can also imagine other particles such as titanium, iron, etc. might be tracked in the same manner. Finally, one of the issues is the possibility for biological uptake of materials. Now, right now, no one has seen any kind of nanomaterials in field-collected organisms, but in the world of toxicity testing, where we want to understand the relationship between exposure and effects, we need a method to not only tell what we dose our exposure medium with, the external dose, but also what's the internal dose. What does the organism actually take up? So we've been working with an approach that uses tetramethyl ammonium hydroxide, that's the molecule on the lower right, and this is effective at digesting the organism. We used to try a few cleanup steps like filtration, which was not very successful. We've also looked at low-gravity centrifugation. In many cases, we can just dilute to the part per trillion levels we need to dilute away a lot of the matrix components. So we've modified this system a bit by adding this tetramethyl high pH system. What does that do to our nanoparticles? Well, we published this both in an application note and in a journal article, which I'll highlight in a moment. And so take a look at that note. But the question is what happens to the nanoparticle? Well, on the left is our silver nanoparticle suspended in water. And on the right is that same particle which was spiked into a tissue. In this case, it was ground beef because of its ease of just going to the local market. And the good news is that not only is the size distribution not altered by the TEMA, but these two samples were diluted in the same manner. So you can also look at the y-axis. And the particle number concentration, the recovery is quite high. So we felt like that the TEMA approach was a viable approach for studying the uptake and transformation of nanoparticles into biological systems. We took this approach and then tried an actual uptake. We used a worm, Lumbricus variegatus. We exposed it to silver nanoparticles. The nanoparticles are shown on the upper right, just suspended in water. After 24 hours of exposure, we took these organisms and put them into clean water to depurate what might be present in the gut. But you can see at the lower left at time 0, the worm before depuration, we can see the particle size distribution and the particle number concentration. As we allowed the organisms to depurate 24 and then 48 hours, we saw a lot of particle loss, presumably those particles which were retained in the gut. But what we noticed is even after 48 hours, there was still a residual of particles remaining in the organism that looked quite similar to the particles originally dosed. So it doesn't look like there was a lot of dissolution in the organism itself. So this has some implications in terms of potential for trophic transfer and a bioaccumulation if some fraction of nanoparticles are not readily excreted by the organism. I think that I can wrap up now as far as these examples I've given. Investigating the transformations and the fate of nanoparticles, as I've shown and both Kevin's work has shown, still a challenge, and we still need to continue developing our metrology. The presence of natural nanoparticles is going to mean our techniques will have to have the ability to look at multiple elements and also have size selectivity, as I pointed out with the single particle, this microsecond single particle analysis. We need further investigation of the uptake and elimination and transformation in organisms, and single particle may be a good approach for this. And by adding these other techniques, either FFF or size exclusion chromatography, coupled with this fast scan, this microsecond scan single particle, helps us overcome some of the challenges such as high dissolved background. I didn't mention the coincidence issue where we tried to eliminate multiple particles entering the plasma. But it provides us with some tools for researchers to investigate this idea of environmental fate and transport. With that, I'll just acknowledge my group, particularly Manuel Montano, who did a lot of the development work in collaboration with PerkinElmer to implement the rapid scan or fast scan microsecond ICP-MS. With that, I'm going to turn it back over to Chatty to wrap up.
Thank you, Jim. I'm going to be wrapping up here shortly. I have a few more slides to talk about. If I want to put single particle ICP-MS in perspective and what it brings to the table. So it takes a conventional ICP-MS where usually the samples are digested and we're only looking at the metal composition or the dissolved composition of the solution. Now we have single particle ICP-MS, which allows us to look at particles at particle level. So we're going to be able to count. So the concentration is going to be the particle concentration or the particle number, the composition of the nanoparticles. We're going to be also investigating the size and the size distribution. In design system, or actually also in natural system, we can use the time information or the broadening of the peak to be able to identify agglomeration. If you want to look at it, what it offers also, Jim just mentioned about the two analyte scanning. So adding to that, single particle ICP-MS will allow us to start investigating the structure of the nanoparticles. As it is in technology that looks at the mass, so comparing it to another technology or visual technologies such as electron microscope, and being able to compare the sizes, we'll be able to come up with information about the porosity of the materials. The last thing that we were trying to investigate is really trying to correlate the shape of the signal to a shape of a nanoparticle. This is something that it's still ongoing. There are some ideas around it, but it's really something in the future that could and could not happen. Along with the environmental applications, we've used the technology to do a lot of other applications. We can investigate nanoparticles in blood for drug delivery or cancer treatment. You can use the technology to look at nanoparticles or sodium oxide or slurry oxide used for the semiconductor industry. So basically, it is a technology that that has been developed, and it has been applied and used in a multiple industry or a multiple type of industry that allows the tracking of nanoparticles and allows the ability to look at the size distribution of those nanoparticles. So with this, today we talked about single particle ICP-MS dedicated for the inorganic nanoparticles. And we would like to invite you to join us on our webcast number 2. It's going to be talking about the axiom TOF, DSA TOF. It's the complementary technique. Think about it as it is a complementary technique for the ICP-MS. So we use the single particle to know the core and the composition from a metal perspective, what's in that nanoparticles, and we're going to be using the DSA TOF to look at what's sitting on the surface of that nanoparticles. Is it functionalized? It's not functionalized. Does it have a PEG? Whatever functionality you want to do, we're going to be trying to guide you about what we can do and what we can see. And in the third webcast, we're going to look more on the quality control of manufacturing nanoparticles, either in liquid, and we're going to be talking about some hardware that allows us to investigate nanostructure at a solid form in an engineered nanomaterials. With that, I would like to thank the audience for joining us this morning for this webcast. With this, I will turn it over to Laura.
Thank you very much. Thank all of you for that excellent presentation. It's now time for questions from the audience. As a reminder, all of you can submit questions. All you have to do is type them in the Q&A box, and you can find that by clicking on the red Q&A widget at the bottom of your presentation window. All right, I'm going to start with two questions for Kevin that sort of go together. So Kevin, this user says, can I use hydrodynamic chromatography to give me similar outcome as FFF?
Yeah, these techniques are really very, very similar. As you might have picked up from the dynamic chromatography, the retention times, at least when you're pumping into an ICP-MS, are much longer. So the FFF certainly has the advantage there. On the other hand, the hydrodynamic chromatography is generally much easier, a lot fewer parameters to play with. So it's a nice technique for that.
Great. And can you also use hydrodynamic chromatography with single particle ICP-MS for speciation studies?
Yeah, with both FFF and HDC, you're not getting a lot of information on chemical species. You're getting information on the particle sizes. It's really when you start working with these ion exchange resins that you're going to get a little bit more information on the chemical speciation. So the nature of the dissolved species that are there. Both the HDC and FFF, where you're looking at those larger particles only.
Excellent, thank you. Next I'll go to one for Jim. If you work at a 0.1 millisecond dwell time, you will also reduce the dynamic range to 2 orders of magnitude. Are there any plans to work on PC detectors to increase their range to, I think this is the symbols are not coming up on my screen properly. I think it says 0.62 milliseconds CPS?
Well, I'm not fully sure I totally understand all the aspects of the question, but I'll try to answer that. The reduction to the 0.1 millisecond has allowed us, again, to really focus on capturing the peak. It allows us to basically chop the peak into a limited number of fractions while removing the background considerably. So go back to the question about the dynamic range. I'm not quite sure I see how that was really related to the microsecond time. We're really more limited in terms of our lower end of the range by the sensitivity of the mass spec. On the upper size range, I think the question was about sizing. We do see eventually we get particles large enough that the signal exceeds the counting ability of the mass spec. In that case, we actually want to go to even shorter dwell times to be able to not go over that. I'm not sure that directly answered the question, but again, we feel like 0.1 millisecond dwell times is a significant improvement over what we've been doing in the past in the millisecond dwell times. If the questioner wants further clarification, they can contact me offline.
Great. Chatty, can single particle ICP-MS differentiate between aggregates and large particles?
To answer this question, there is a few things that I've been noticing running a few and multiple projects. Bigger particles, the transient time of that pulsation is not changing a lot from a 50 nanometer pulse. If I'm going to give you more of a put it in perspective, if I run a 50 nanometer particles, the transient time for that pulse is about 300 microseconds, and if I run 100, it's probably around the 320 microseconds. It's not like way larger than that. But when you have an aggregate of 2 or 3 50 nanoparticles together, the transient time of that particles is in the 600, 700 microseconds. So yes, the question is, you can use the time-based information from that transient time to allow you to know if it's an aggregate, if that particle or that big signal is an aggregate or a large particle by itself.
Excellent. Kevin, I'm going back to you. What is your opinion on the use of UF techniques like Amicon filters to remove much of the dissolved fraction in order to reduce the background signal?
I can't see how that would help, simply because with the Amicon, what you're interested in is getting the retentate, and in the retentate you'd have less and less dissolved. The columns that I was showing you, you can go directly online, so you're pumping your sample through the columns and looking at the particulate and retaining the dissolved. So it's sort of the opposite of what's being proposed there. Though I really encourage people to try all sorts of columns. There are probably lots of columns that we've missed. We're just starting this research, and I'm sure there are lots of ways to remove the dissolved. But ultrafiltration techniques, I don't see how that could be done easily. I can see how that can be done, but I don't think how it could be helpful easily.
Okay. Jim, does there currently exist any sediment digestion method to determine silver nanoparticles in sediments? And if it does exist, what is the method and which chemicals are used for the extraction?
Right. So I have not seen anything in the literature yet specifically in this area, but in our lab we tried the tetramethylammonium hydroxide approach on some soils. We got lower recoveries. In the case of tissues, we were getting something like 90% or better. For the soils, we got about 50% or so. But we also were happy that, again, we did not see any real transformation when we spiked a known particle size. But right now, I don't believe that there's anything standard. Folks have tried something simple like, again, water and surfactants and sonication. But certainly acid digestion is not going to work. But the TEMA does seem to be reasonable. But again, I just encourage whoever asked the question that this is an open question and it needs to be investigated more.
Great. Chatty, do you have any white papers for applications in pharma?
Yes, we actually have one that is already published about the ability of tracking nanoparticles in blood and urine. We are currently preparing another one that talks more about investigating cancer drugs that have gold nanoparticles in the middle and different functional group on the surface. And we're going to be able to track how they do behave in the blood and urine, and we're investigating the residential time in the bloodstream, and we're actually looking about if they would dissolve or not. So what I can say is keep an eye on PerkinElmer website. There will be many applications coming down the pipeline.
Great. Jim, for the worm study, you found some residual nanoparticles in the organism. Did you do any toxicity studies to evaluate how the presence of those residuals could affect the organism?
We've done other tox studies where we've looked at other organisms, but in these particular cases, we were interested in accumulation and exposure. We expect, in the environment that it's unlikely, I think, that we'll see direct toxicological effects in the initial organisms because the concentrations are so low. So we specifically worked at sublethal concentrations to try to track the accumulation. But certainly we've done some studies with aquatic species, Daphne magna, where we've taken the concentrations up sufficiently high to see mortality. And so you can take both approaches. Again, our interest was in exposure and not toxicity.
Great. Chatty, how long does the analysis take using single particle ICP-MS?
Well, it all depends on the particle concentration or what you're trying to achieve from your analysis. Ideally, in most of the application nodes that we're currently running, we use a minute or so. Sometimes if we have a certain particle number that is high, 30 seconds is usually sufficient to get you a proper statistical distribution for your nanoparticles. But if I want to put on it a number, I would say really it's definitely less than a minute. In some instance where you have only few particles in your solution and you want to build the statistical information about the particles in that solution, so you might want to acquire data for a longer and extended period of time. But really, it's usually less than a minute.
Fantastic. Well, unfortunately, we're out of time, so we're going to have to wrap up. I thank all three of you for your excellent presentation and for answering those questions.
Thank you very much.
I'd also like to thank everyone in the audience for participating today, and I'd like to thank our sponsor, PerkinElmer, for making today's webcast possible. This webcast will be available for on-demand viewing through November of next year. You'll receive an email from Spectroscopy letting you know when the webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye. Hello, everyone. Welcome to today's live broadcast, Single Particle ICP-MS, and its advantages in analyzing nanoparticles in environmental matrices. I'm Laura Bush, the Editorial Director of Spectroscopy, and I'll be your moderator for today's event. We are delighted to bring you this web seminar presented by Spectroscopy and sponsored by PerkinElmer.